Hello and welcome to our last topic that we're going to talk about. This is our option. So when you are taking your IB exam, the option is its own paper. So paper 1 is multiple choice questions spanning all of IB biology. Paper 2 is like DBQ short answer questions. And then paper 3 is all about the option. So this is the first sort of intro into our option, which is neurobiology. We're going to start with neural development, which is how basically your brain develops, your spinal cord develops, how your nervous system starts out. So how does your nervous system actually start out? So this is like when you are in the womb, like a fetus. This is how this starts to work. So you have this is like the first thing that forms when you are a fetus. And so you have the notochord, which is going to be a key player here, and the mesoderm, which is another key player here. And then you have the ectoderm, the neural plate border, which is this green strip, and then the neural plate. So the cells located in this neural plate are differentiated cells. Eventually, this neural plate border separates, well, not eventually, but it does separate the ectoderm from the neural plate. The two neural plates will eventually bend up. You can see this like almost like V shape start to form, and the neural plate borders will join, and they are no longer called neural plate borders. They're now called the neural crest. And so it starts off as like a little like almost flat line, and then it will turn into a V. You can kind of start to see a shape forming. I think you'll recognize it on the next slide. And there you go. Look at that. It totally forms together. The neural tube disconnects from the neural crest and from the ectoderm. So now you have like distinct layers. You still have your notochord and you still have your mesoderm. Those are still present. They just haven't had like a chance to do anything just yet. The neural crest cells, which again are this like green strip. It's not green in real life. This is just to help you. These cells differentiate to form most of the peripheral nervous system, and that's basically anything that involves nerves that is not your brain or spinal cord. So peripheral nervous system would be like any nerves that are in your like fingertips or anything like that. Okay, the notochord will degenerate. So this little like tiny thing at the bottom, and persists as the nucleus pulposus, which is the inner core of the intervertebral discs. Those are the discs that go in your spinal cord. And then the mesoderm, which are these like pink, well, they're like red in this picture, red egg-shaped things, will differentiate into somites. This is how you spell somites. I don't know. It just is showing up with the red line, but it is how you spell it. They will differentiate into somites, which are the precursors of the axioskeleton and skeletal muscle system. So all of this is like the beginning. You'll see that as these continue to develop through fetal formation, that like your crest cells go lower and become the spinal ganglion. That is the nerves that extend outside of your spinal cord, which are beyond your spinal cord, which is why they are part of the peripheral nervous system. But you can see how this like really develops into what we can kind of visualize as our vertebrae. So all chordates, all organisms that have a spinal cord, develop a dorsal nerve cord through two-step neurulation. So primary and then secondary. So primary is the neural plate creasing inward until the edges come in contact and fuse. That was like the first slide. And then secondary is when the neural plate forms a cord-like structure that migrates inside the embryo and hollows to form the tube. So you have that neural plate coming together, right, and forms almost like a circle, but it has a hollow inside, and inside of that will allow your spinal cord to develop. The neural tube is the precursor to the central nervous system, the CNS, which is your brain and spinal cord. So the neural tube is like the embryo fetus version of your spinal cord. So initially, neurons are part of the neural plate. They are called neuroectodermal cells. They're just like baby neurons, right? They eventually develop into the nervous system. When the neural plate forms, the neural tube, it eventually differentiates all of those cells into functioning neurons. The specific cells that develop into neurons are called neuroblasts. So neuroectodermal cells are your like baby cells. Eventually those will develop into neuroblasts, and those neuroblasts will develop into what you know and will love, neurons. And the pictures up at the top of these like crazy branched cells, those are neurons. As you continue to develop and cells continue to divide through mitosis, cell development continues as the central nervous system develops more formally into the brain and spinal cord. Remember, this is like when you are in the womb. This is like embryo fetus. So this is like really, really early on in your life. Now, we've been talking about how things would typically work if you were to have a typical development structure. However, sometimes that doesn't happen. And when your neural tube doesn't actually close, you get a disorder called spina bifida. So when you are developing, the centrum or the body of on the ventral side of the neural tube, the body is developed. And then these cells form tissues that migrate around the neural tube and eventually form the vertebral arch. Okay, so this like arch here, you can see it in the top picture. So here is your like notochord, and then you have your arch forming. However, sometimes these tissues don't meet up and they don't fuse together, and so you have this gap, and you can see this on your second picture, the bottom picture. And so you have this almost like an opening. It's an unprotected area of your spinal cord now, right? This blue thing has become your spinal cord. You have nerves coming out of it, and now it's unprotected. And if you were to reach right around right now onto your back and feel your spinal cord, you can feel the vertebral spines. That's what's sticking out. It feels like bumps as you go down, not like extreme bumps, but you should be able to differentiate the different spines. And that's what's missing in spina bifida. So when they don't fuse together, what's formed is spina bifida. The severity can range from mild to debilitating. It depends on how much or how little is missing from that arch. So there are two types of cells that can develop from the neural tube. Neurons, which we've been talking about that for a while, and glial cells. So neurons carry messages, and glial cells provide physical and nutritional support for the neurons. So neurons are like your MVP player, and glial cells are like your water boy that makes sure that your MVP can like do what they need to do. So glial cells provide a scaffolding network where neurons can migrate to their final location, mature, get ready to send out their axons and dendrites. That's that branching aspect on a neuron. And so glial cells are vital to the production of neurons. So what's interesting is in brain development, as some neurons are produced, they'll get produced in one part of the brain and then migrate to a completely different spot, and then that will be where they basically live for the rest of their life. Remember, neurons don't really travel that much. They send messages from neuron to neuron to neuron, but they're sort of just like existing. They also don't really develop anymore as you grow like outside of the womb, as you like if you headbang a lot and you kill a lot of your brain cells, they don't come back type thing. But anyways, the idea here is that immature neurons, as they're developing, will start in one specific area depending where like those glial cells are that can support them, and where they end up could be somewhere totally different. Now, I've mentioned axons, I've mentioned dendrites. Let's talk a little bit more about like what they are and how they grow from these immature neurons. So immature neurons consist of just the cell body and a nucleus, of course. All eukaryotic cells have a nucleus, and cytoplasm. Then axons grow out from the cell body, and at the tip of it, they create what's called a growth cone. So chemical stimuli will determine how the axon differentiates, how it grows, in what direction it grows, and then as they grow, they will synapse with their target cells. So they will make a connection with whatever cell they have been tasked to interact with, whether it's the tip of your finger so that you know to bend your finger, or if it's your eyelid to know that you are sending a message to blink. Whatever it may be, the axons will eventually synapse or make that connection with their target cells. Now, some axons extend beyond the neural tube towards the target cells. So they have to extend out of the central nervous system. Remember, the central nervous system is just your brain and spinal cord. And they form circuits with muscles to control voluntary muscle movement. So this is you actually choosing to move something. So moving your arms up and down or kicking your legs, right? So the muscles attract neurons by producing a chemical saying, hey, I need an axon to come hang out with me, and I'm producing this chemical to lure you over here. Okay, those chemicals are called CAMs, and they activate enzymes in the axon that cause that growth cone to grow towards that muscle. Okay, so the muscles are very sneaky in this, where they, well, not sneaky, but they definitely lure neurons out from the spine, because neurons are content where they are, but they will be lured out from these chemicals because the enzymes in the axon are like, yes, I like that, I want to go there. Now, the longest neuron is the sciatic nerve, and that is all the way from your spine to your big toe. It is a huge nerve. That is very, very long, and it goes like kind of in your groin area all the way down. You may have felt sciatic nerve pain before. It feels like you like can't lift your leg up because it just like has like a stinging pain. Usually it's due to something else, like well, like pregnancy can do it. You can hurt it accidentally if you're like doing too much or stretching in ways that you're not really used to stretching. Sometimes you can just sort of bruise it a little bit, and then you'll feel some pain there, but it will heal. Now, I keep saying that like, oh, you form this connection with a target cell or a muscle or whatever. You can form it with like multiple cells. So as neurons develop, they can form multiple synapses with neighboring nerve cells and other target cells. So it's not like each neuron has one cell that they target or one muscle that they target. It could be like 10, 20, like many synapses they can make connections with. They obviously need to be in the same general location because neurons aren't huge. So they will but they can make connections with multiple things. So neurons will keep trying new connections until compatible connections are made. So they like to make connections. I mean, there's so many branches on the axon, so they really can make tons of connections. They like compatible connections, of course. They do, however, or they will, however, try any sort of connection, and if it's not compatible, then those connections will just be eliminated. When the compatible connections are made, the axons develop synapses. The smallest number of synapses can be 2, but most develop more than 2, even hundreds. This allows complex communication. This is why you can do so many things at the same time, even things that you're not thinking about. Like, as I'm talking to you, my hands are waving because I always talk with my hands. My eyes are blinking, I'm breathing, I'm sitting down, so some of my muscles and my digestive system are doing involuntary movement. All of this is happening, and my neurons are making all of these connections for me, and I probably have hundreds, if not thousands, of neurons doing work right now in my body, and they're doing it like seamlessly. I'm not thinking, well, now I'm thinking about it because I'm talking about it, but I'm not like actively telling my neurons to do anything. It's very complex and it's very interesting. Now, you make tons of connections. I mentioned that neurons like to just sort of like reach out, make as many connections as they can. Some are favorable, some are unfavorable, but a lot of the synapses that are formed during early development, if they're not used, they'll just start to disappear. So this is why, like, you never forget how to ride a bike. Like, even if you don't do it that frequently, you've made such a strong synapse connection with how to ride a bike that when you get back on a bike, you might be wobbly for a minute as your synapses try to like reconfigure. Whoa, I haven't done this in years, like, how do I ride a bike? But it'll come back really easily because you've already made those connections. Okay, the more frequently you use a synapse, the chemical markers that are in your body will just cause that connection to be strengthened. So if you are like typing all the time and you are like, I don't know, doing something on the computer, eventually you'll be able to do it with your eyes closed, or like you hear people say like, oh, I can do that in my sleep, because you've done it so much that your brain has basically automated the process. So anyways, again, if you use it, it's strengthened. If you don't use it, your body will just eliminate it. There's no reason to keep that if you're not using it, because it's just a waste of energy and time. When you eliminate these neural connections, it's called neural pruning. So pruning in like a gardening term is like cutting the excess off of like a bush or like a tree or something like that. And so you're basically doing the same thing here. You're cutting the excess off of your axon. So plasticity is an interesting concept. I think it's probably the coolest part of our brain. It's the ability of the nervous system to change and rewire its connections. So this allows growing of axons and dendrites, new synapses being formed, elimination of other synapses, pruning of dendrites, which are those branches of axons, even whole neurons you can prune. And this is all based on how individuals use their nervous system. This is how we form new memories. This is for some types of reasoning. This is for people who suffer a like damage to the brain, like a stroke. Your brain can just rewire itself to, like, okay, there's a part of my brain that has damage to it. It's not working the way that it has worked previously. So your axons and your neurons and your dendrites and all of that will just sort of skip that part of your brain that isn't working, go around it, and start rewiring itself to still have the same outcome. You're just skipping or taking a new road to get there. It's very cool. And it's really helpful for those that need this. So this is kind of what I was talking about, a little bit more in depth about how strokes can encourage reorganization of the brain. So typically a stroke is blocking or disruption of the blood supply to a part of the brain. Usually it's a blood clot, or you have a rupture in your blood vessel, so you have bleeding of the brain. So this causes part of the brain to be deprived of oxygen and glucose, and if you don't have cell respiration taking place, right, oxygen and glucose are the reactants of cell respiration, then you don't have any energy being formed, those neurons aren't getting what they need, and they end up being damaged beyond repair and eventually will die. The recovery, though, from a stroke is possible because of the plasticity in your brain. So new parts of the brain will take on new functions, or different parts will take on new functions, and your remaining neurons will rewire themselves to match up to these new areas of the brain that are taking on these new functions. So, very interesting. We'll talk a little bit more about the development of your nervous system, but really we're going to start jumping right into all of the bits about your brain, the parts of the brain, and kind of how your brain works. So, good job, and I'll see you next time.